Okay, good evening, ladies and gentlemen. Thanks very much for attending this sharing section. And also, we thank DIY BioHK for inviting us for this sharing. So, specifically, we are from HKUST RISE iGEM team in 2015. And today, I'm Eric, I'm Edward, and this is Eric. We will be giving you a brief sharing about what synthetic biology and also what iGEM are, and how synthetic biology has led to a foundation of iGEM, and also we'll talk briefly about our project, and lastly we'll try to show you how synthetic biology and iGEM are connected to our society. So what exactly is synthetic biology? So it's been an emerging field in the recent decades, and it's a very broad and diverse field in today's study. And it's very different from the traditional biology because it does not only require what a protein does or like how the pathway goes, but it also requires us to know a lot about knowledge on the physics, mathematics, engineering concepts, and we harness all of these subjects, the power of these subjects, and to study and create novel biological systems. And actually it has attracted a lot of scientists to work in this field, and it has raised a lot of interesting questions. For example, we have one big topic, the synthetic genomes here. It is about, actually a few years ago, there's a scientist who has chemically synthesized a set of genome, and he tried to put it in an isolated cell envelope. And actually, this kind of semi-artificial cell has exhibit the living characteristic of microorganisms. So it's a big leap in the on the area of creating artificial life. And also, with synthetic biology, you can produce different kinds of industrial enzymes, because, for example, you can produce a lot of enzymes that could clean the stain on your clothes, and also you can produce some enzyme to digest some polluted substance in the river, like the oil spilled. Also, lastly, just one more example about the information storage. You can never imagine you can store information in our living cells, right? But actually, a few years ago, also a scientist has developed a method to store information of entire book into a genome. So, which is also some interesting way of doing synthetic biology. So, actually, this emerging field has not only attracted scientists, but it also attracts a lot of engineers to work in this field also. So that's sort of leading to the foundation of iGEM. Why? Because actually, the three co-founders of iGEM are all engineers. So actually, none of them has biology backgrounds to begin with. For example, Dr. Andy was studying civil engineering for his bachelor degree. Also, Dr. Knight has been working for some computer science, so it's not related to biology at all. But then they found out that the powerfulness of synthetic biology, and they start falling in love with it, and then so they start working in this field, and later on found the iGEM Foundation. But what exactly is iGEM? So you can actually see it as a community or a competition where people come together, or students specifically, they come together to work on a summer project related to synthetic biologies. And here are a set of the core values of what we treasure in iGEMs. For example, the education. It is all about learning experience, where we have to take initiative to learn everything by ourselves. We learn how to do a lab work, how to conduct a research process, and we try to find our way to approach scientific questions. And also, we have learned a lot of other things like codings. We have to make your own wiki page. And also, we have to learn to approaching people for more information, because we have to contact others for their opinions. Also, we have paid a lot of attention on the safety and security issue, because nowadays safety and security is one of the main main questions we have to ask about when you are doing your projects in synthetic biology. Because with synthetic biology, you can basically create any kind of cells you want. You can create a kind of cell that are very pathogenic, that might be harmful to your environment. So even though a student may not be intentionally doing that, but what if he just released it by accident? That's why we need to properly address these kind of questions, safety and securities. And also we treasure a lot about the community and sharing. So iGEM is an open community where we share our knowledge, and specifically through a thing called standard biological parts. So this is some standardized genetic elements where other members in this community can easily use them and assemble whatever kind of genetic circuits you want. So which can be really powerful. And we have different kinds of categories in iGEM. So we refer to tracks. And for example, we have the environmental tracks. In 2014, a team from Taipei actually conduct very interesting projects, which is about solving the problem of coronary disease disorder. So this kind of coronary disease disorder is the main cause of causing the massive death of bee populations, which may be really detrimental to the environment. So they found that it's about parasitic worms inside a bee. So they try to engineer E. coli to kill that kind of parasitic worms, and then they successfully demonstrate this kind of phenomenon. And also we have the bioenergy, and also this one, the food nutrition, where also is one of the most popular topics in iGEM competitions. So our team from Calgary actually conduct a project called the fairy tale, very fancy. Okay, and but what it is actually is to sense a kind of substance called ferritin, which is present in pathogenic E. coli. So they developed a method to sense this kind of foodborne pathogens in the food, in the water, and anything you can consume. And also another very useful project, which is conducted by Utah State. They have actually carried out a project on the spider web. So as you may know, spider silk is very useful in terms of its strength and also its flexibilities, and you can even make it into the bulletproof vest with the spider silk. So they try to put the spider silk making protein into a E. coli and try to form sort of like raise the E. coli and harvest a lot of spider silk. But apart from those, we also have therapeutics, which is about medicines and a way to a kind of treatment. And also we have measurements, and we have we even have software teams where a lot of China team and Indian team will participate on this category because they are really good at coding, and also they are really good at doing modeling. So modeling is also a very big part in iGEM, because in iGEM, as I said, we just not rely simply on the traditional biology, but also we use the power of computational power and also the mathematics to represent. We use this kind of mathematical equations to represent a biological system. We simulate and then we predict what we want. So here comes our project. So back in 2015, we have actually done the project called potassium, phosphate, and nitrate biosensors. So by its name, it suggests that it's a living sensor that senses these three kinds of substance. And in that year, we have a quite unique experience, because this is the first time in iGEM history we have two teams in two different continents have come together to work on the same project. So actually, these guys from the right are from Rice University in Texas, And then they come all the way from US to Hong Kong University of Science and Technology, and we work together in the lab, and we discuss on what we do for human practice and so on and so forth, which is a really great experience for in terms of collaborations. So again, this embodied one of the core value of iGEM, which is the community and sharing. In a community, we come together, we nurture ideas, and we work together to achieve the common goals. So let's come back to our projects. But why do we want to work on this kind of project? Is because when you are doing farming, one of the main issues we have identified is that the plant, almost every plant, needs these three kinds of macronutrients, which are nitrogen, phosphorus, and potassium. So deficiency in any of these kind of macronutrients will lead to poor plant growth, which will be a really huge problem for farmers or even the greater populations. So because people cannot get sustained supply of food. So we try to come up with a way to monitor the level of this kind of nutrients in the soil, so that the farmer can counteract the situation where deficiency of any of these macronutrients in the soil. And in the following times, I'll pass it to Eric to talk more about our projects in detail.
Okay, thank you. So now we have our problem here, which is we want to know the amount of the three macronutrients in the soil, but how do we do it? We tap into the endogenous sensing system in bacteria, and we use the E. coli to do the job for us. So in the nature, E. coli or any bacteria will have to have a way to sense their surrounding for nutrients. For example, potassium ions. It is important for their survival because the fluctuation of this ion could be fatal to the bacteria or any other organism, and therefore they have to have a way to sense the amount of these iron in their surroundings. And specifically in E. coli, they have a set of genes called KDP, which responds to the change in the potassium ion. So they have a channel here in pink, which imports which allows the potassium ion to go from the surroundings into their cell, and then you will stop the phosphorylation. Phosphorylation is the activation of some other protein, and ultimately the stop of the phosphorylation will lead to the activation of some other gene, so that the bacteria will know that the surrounding is lacking of potassium ions, and they will make some protein to react to this, and so that they can move away from this area, or to make more ion channels so that they can absorb more potassium ion into their cell for their survival. And we tap into this kind of system which responds to the surrounding ions level, and we use it in our project. So now we have the power, or we have the set of genes that allow us to monitor the ions level in the surroundings. And similarly, we've done the same for phosphate ions and nitrate ions. But now we have the tools to sense it. The bacteria will know how much there will be, but we need ways to report it, so that humans, we can know from looking into the bacteria and read out how many ions are in the surroundings. And that's what we did. We put the green fluorescent protein from jellyfish and into the genes exist in E. coli, and at the end when the ion channels open or close, there are fluctuation in the ions level, and then the reporter genes will be turned on or turned off. At the end, we'll read from the fluorescent signal, and then we can tell how much ions is in the surroundings, is in the medium surrounding the bacteria. Now, you can see we put 3 different colors of fluorescence protein for each kind of macronutrient, so that we can tell not only 1 potassium, but 3 potassium, phosphate, and nitrate ions in the surroundings. And what we want to do is that with this biosensor, we want to tell when there's a deficiency in the ions level, right? Because the problem is that when the plants are lacking the ions, they will have some plant disease. So we engineered the bacteria genomes so that when there is a fall in the ions level, the light actually goes up. As you can see, when the potassium ions level decreases, the light signal increases. And that is exactly what we want to do. But building these 3 sensors is just part of our experiment. We need a way to combine them and make sure that even when combined, they are still working they're still working as individual sensors without much interference. And that's what we do next. We put any of the 2 sensors together, and then we measure if the output will be affected. And indeed, we found that there's a significant change when we try to put 2 sensors together, and we compare with the single sensor genes, and then we find that there is a problem. So as you can see, the green line here is that it's a single output construct, so that there are only 1 biosensor, and in the blue line there are 2. Immediately you can see that when there are 2 sensors working together, the signal decreases, and this could lead to some of the this effect could potentially renders the system dysfunctioned. But so far we identified this problem, but we do not have the time to solve it yet, and we are waiting for maybe future iGEM teams to pick up this project and work it to further engineer the system so that it can prevent the same problem appears in the future generation of this system. But yeah, so we have identified the problem, and we did not conceal this, but instead we show it to the communities and hope that some other teams will pick up the project and work it further. But laboratory is just part of the iGEM competition. The other side of the iGEM competition is to face the society and get feedback from the society, and at the end, based on their opinions from the society, we engineer our project to the way that the society will accept it, and the science and technology will benefit the society at the end. And this part of the competition, we call it human practices, because the ultimate goal is to build a better society using the synthetic biology. As you can see here, scientists have some technologies and scientific knowledge, but they have to listen to the society get their feedback and then further improve on their technology and science. And in our case, our human practice, we do several things. What we really want to do is to get the feedback from the societies on our project. So our project is to engineer biosensors, right? We want to alarm the society that we have these biosensors. What are your concerns? We want to get their feedbacks on the appropriateness of the setting. So is the bacteria a suitable choice, for example? Is there any risk using the bacteria as a biosensor? Is this useful as a biosensor? And is it sustainable using bacteria as a biosensor? And at the end, we do interviews on local farmers, and we get their feedbacks. And for the first three items, what we gather from the local farmers are that they think that using bacteria, living bacteria as a biosensor that may potentially put onto the field and on their crops is not very appropriate. It could raise some security and safety concerns, and it may not be as useful. And therefore, we think of other ways to solve these kind of concerns. The first one is we can engineer a paper-based lysate, a system which contains our biosensor. So which means that instead of using a living bacteria, we are trying to use some cell-free cell lysate, which means that we destroy the cell, we destroy the bacteria, and then so that it can no longer sustain itself in the environment by itself, but it retains the ability as a biosensor. And in such a way, the safety and security concerns can be eliminated. And it could be it could be applied to the field more easily when we are using this paper-based lysate. So it could be one of the ways that we could further improve the project. And on the other hand, only being our project engineered a biosensor, but having only the sensing abilities may not be enough. So they also suggest that maybe we can engineer this biosensing so that when it detects the lack of nutrient in the surrounding, it could actually synthesize something that the plants can use, so that as a rescue, when there's a deficient in nutrients, it immediately rescues the plant. So that is two of the ways that we heard from the community, from the society, and two of the ways that we can further improve on our project. And over in Texas, they also try to apply their knowledge into helping to build a society that are good at STEM, science, technology, engineering, and math. And so they initiate a bioethical debate in the high school in Texas, so that the high school student can debate on some of the bioethic question that commonly found in synthetic biology. So in such a way that our members in Texas, in Rice University, they share their scientific knowledge and to the community, to the society in their state. Okay, so that's our iGEM 215 competition. That's what we did. But you may ask, other than iGEM, what can we do? How can we get involved in synthetic biology? There are many others synbio organizations. And for example, we're here because DIY Bio HK, and it's a good way to start doing some biohacking and to get involved. And you're here because you want to get some information about synthetic biology, and I hope you get some of the knowledge and some of the insight we encountered in the competition. But I digress. Last year there is a Hong Kong high school, UCCKE. They joined iGEM as a high school team, and they did pretty well. In the first year they get a medal. And there are, other than iGEM competition, there are also biomod competition, which is also held in the state. That is highly related to engineering biology. And there are other startups spin out from iGEM competition. For example, Benchling. Benchling is a software company that aims at accelerating doing the process of doing biology. So in the past, editing a sequence may be troublesome without a suitable tools. And the available bioinformatic tools are scattered around the internet. Benchling helps to integrate all of the necessary software into one single place, and so that the scientists can accelerate their progress. And Autodesk has been a very supportive sponsor of iGEM competition. They want to promote this kind of movement in synthetic biology. And there are also PVP Biologics. They aims at engineering enzymes to solve one of the disease where people cannot digest gluten. And there are many ways that public can engage in synthetic biology other than iGEM. Maybe public can join some conference, SynBio Beta, a highly many synthetic biology startup actually went to this kind of conference and consortium, and to work with other company, and at the end they gain some and they share their knowledge. And maybe at the end you can also start a iGEM team in Hong Kong, maybe in the coming years, or in the coming years. So that's it for our sharing, I guess. And thank you.
Any questions, anybody?
Can you tell us a little bit more about how you engineer the genome of the E. coli to make it sense the deficiency in nitrogen? So how do you actually go about the mechanism? How do you actually go about doing it?
Okay, I think so I actually skipped a lot of details in here, right? So to engineer this biosensor, what we need is to have some of the system that naturally occur in the bacteria, and they have some protein that are sensitive to the potassium level, so that when the levels are below a threshold, it will be phosphorylated, so a phosphate group activate. And with this kind of signaling, it actually some of the phosphorylated protein will act as a transcription factor that initiate the transcription of some of the gene. And this is what we want, right? When there's a change in potassium ion, there's a change or activation in their genes. And to build a biosensor, we put our reported gene under the control of this system. And so that when the change in the potassium ion or any ion can be reported through the reporter. So at the end, the change in the ion level result in the change in the expression of
So
our reported gene.
It activates a transcription factor.
Yeah.
It triggers, there's a transcription factor. It turns the genes on.
Yes. And this case apply to all three. So they are using similar signaling pathway, just using different protein to sense different ions.
So they produce a different protein to sense the
Yes,
pathways.
yes, because the ions are very important to their survival. They have these mechanisms.
So the potassium phosphate in the nitrate sensor, is it already present within the E. coli, or did you have to engineer that pathway also to detect the potassium phosphate in the nitrate?
Oh yes, the pathway originates from bacteria, and the bacteria happens to be E. coli. And we didn't work too much on the phosphate sensor because previous year some teams already engineered some of the promoters and their genes that are related to detecting phosphate. The same goes to nitric.
Is that because E. coli's got a fewer genes, that's why it's so easy to manipulate?
I would say it's not correlated because E. coli is a well-known model organism, so there are a lot of established techniques about E. coli. So that's why people can easily handle them. It's not about the number of genes.
Okay.
Yeah.
Also in iGEM we want a bacteria that grow very fast because it's a very short period of time, and it happens to grow very fast, E. coli, and so that we use it in our competition.
So after the results came out, so for example, there's a change in potassium, the transcription factor. Let's presume that it produces a protein. So were you studying the protein level changes in the thing? So do you guys like use the phrase or something to study the other changes? Like how did the mechanism of detecting the thing work?
Yes. Okay. So the question is, how do we tell the change, right? We choose to use some fluorescent protein. So GFP, RFP, and BFP, they give different colors when they receive UV light. They give out fluorescent color of in different spectrum. So we choose to use fluorescent protein mainly because you can use your eye to see it directly on the plate. You don't need to use a very specific instrument. You just need a UV light source, and for the case of RFP, you can see it with bare eyes. So it's very convenient for us to when we are doing the experiment.
So how do you know whether it's deficient? Because then, so the color is higher when it's deficient. So what is your standard? So when it's sufficient, what is the color, and how do you know what is the standard marking of the color? So how do you tell whether it's deficient?
Between the concentration of the ions and the intensity of light, is there any standard relationship?
Yeah.
Okay, so that is what we actually want to find out in our during our years in 2015 iGEM competition. And what we really want to do is, first we want to have we want to find out this kind of curve in the first place, so that we know that it is responding to the change in the ions level. The first thing we will need to do that, right? So that we show that it is responsive. Now to the problem of sensitivity. So what is a deficient level? We want to find out this. So we want to look at the literature, and we look into how much, what is the normal potassium ion level, and we want to compare it with the chart that we have, and so that we will know that if our reporter are giving out this amount of signal, it tells us that the soil or the sample contains this amount of ions level, and then we will know that, oh, if it is brighter than this standard, this normal sample, it means that something is seriously indeficient. So at the end, we will need to have a standard, but at least first step is to show that our system are responsive. Yep.
You were saying that when you try to combine two different currents in the same E. coli, but it didn't behave when they were individual. Do you have any theory or hypothesis about this like that?
In fact, we expected these kind of phenomena, and therefore we do this measurement. One of the possible explanation is that the system are using too much of the bacteria's resources, right? So the load of our biosensor exceed the capacity of the bacteria. So we're overworking the bacteria, and therefore it gives less protein at the end. And that could be one of the theory. The other theory is that there are interference in our biosensors, but we cannot prove it at that time because we are running out of time, and so we couldn't prove that problem. But that are the two possible ways to explain the difference in the expression patterns.
So how do you suggest that, so for example, when you did the human practices, you got a few inputs and feedback from the farmers, and you said that you would be using cell lysates, but one of the essential things over here is that you need a pathway so that the genes get transcribed and the protein is generated in the first place. Then how would you be able to do it if we had like a cell lysate where you don't have a pathway at all?
Actually, in the lysate, they will contain the necessary components, and if they don't, we'll add it to it. Okay, so what are the necessary components that are required for our biosensors? They are simply proteins, right? So if, for example, our lysate do not contain these kind of proteins, and therefore if the system do not contain this protein, the system won't work, right? So we can simply purify some amount of this protein and then put it into the lysate. Because it's a lysate, we do not care about the membranes, the boundaries. We put it in and it will contains these kind of proteins, right? And is one of the benefit if we switch to use a cell lysate. It removes the boundaries and it makes a lot of things, it simplifies a lot of processes.
One thing that you said was that, so you've worked on this project now, up to now, well, up to the end of the competition. And now you hope somebody else will take it up again. But why aren't you continuing with the work? You're still at the university, you're still there, the pieces are still there. Why put it down for somebody else to work on?
Yeah, it's a question that puzzled our team for a very long time. It seems that HKUST team are very ambiguous. Every year, new members join the teams, and they are ambitious to have their own project, to work on something new, something novel, and I admire their curiosity to try on new things, but it only makes sense when a project is continued over a period of time until it reaches the answer. And that is something that we try to do. Karen in here actually try to solve this co-expression problem. And yeah, we are slowly improving our mentalities to not try to have something new, but something with quality. Yeah, that's what we try to do every year, but ultimately depends on the members what they want to do at the end. If they don't want to continue, and maybe we can only rely on other teams to pick it up.
Yeah, but as I understand, so if every time you sign up, you get the box of blocks. You know, so you still have your box of blocks from when you signed up from your competition. So can't you as a group just continue with that on your own? Continue with the project. Why do you need?
But that also depends on our personal life. So because that
What about funding? You probably do not get funding.
Oh, we are lucky teams because like Dr. Chow has taken care of it for us. And so what he wants for us is that you focus on learning. So they like he wants us to maximize our learning experience. Just that what you need to do is take initiative and learn. So you don't need to care about the money.
So UST gives funding for projects like this?
Well, I think Dr. Chow has to look for like not only just from UST but also like sources from outside UST. Yeah. And by the personal life, I mean like since we are college students, so actually after the first year we have like increased our workload for our academic life, so we may not have enough time to take up the project again. Going holiday, yeah, we can think about that, but
if you're really passionate about it.
yeah, that's cool. Oh, actually, following this year, so in 2015, the Rice University sent someone over to UST to do some research. Actually, on the next year, we also sent few guys over to Rice University to conduct related research.
So we are lucky team. We don't worry about funding.
Yeah.
So how did you convince another university, or how did they approach you, or did you approach them? Like considering it's like a
Oh, you mean with Rice University? Actually, it was like bridged by the Professor Chow and one of the professors in the Rice University. So actually they met in the iGEM competition and they become friends, they talk a lot, and then they think about like maybe make a strategic alliance between two schools. So that's why they come up with this kind of collaborations. Yeah. And also actually not just we don't do collaborations only in this kind of like cross-continental like joint team things. But actually, over the years, we have been doing collaborations, just that in a like remote manner. So we don't really contact in person. We don't really come together in person. we contact by email and see if what we can do on like each other's lab separately. Also as a collaborative work.
And also for last year high school team, they actually approached several existing iGEM teams in Hong Kong, including our teams, and I think they also contact someone in HKU iGEM team and ask for their help, maybe sharing, maybe cloning, and maybe ask for some bio breaks. And yeah, it's a community that is growing.
Questions? No? Well, thank you very much. Thank you. So, oh yeah, thank you.